right. Good afternoon, folks.
Good afternoon.
I can kind of see how everybody's feeling today based on the chat. All right, so I'm gonna start with announcements as usual. And unfortunately, the first announcement, it doesn't make me happy. And that is that Ventura College is going to stay remote until at least mid-February because of the continuing high rate of COVID infections. So faculty are talking amongst each other, trying to talk about what are we going to do about the labs that are scheduled to be in person, but are now not. Missing the first 2 weeks, that wasn't really a big concern, but now we're starting to get to a little bit more time. So I have set up a poll for you to anonymously express your opinion. And what would you prefer to do? Okay, that's everybody. No surprise there. Most of us would rather go in person. That's good. It gives me a place to start in my conversation about this topic. I'm not sure if we're gonna have that as an option, but I will push for that, since that's what the majority of us would like to do. And we'll just kind of have to see what happens. Does anybody have anything they want to say on the topic? Nope. Okay. Based on how we started this semester, is it going to continue for the whole semester? I do not think that that will be the case. I think that we will be back to face-to-face labs. We are already starting to see the numbers of new cases level off. More in the eastern part of the country than out here. They kind of started the surge before we did, but I do think that we're going to level off and cases are going to go down. And when that happens, we'll be back face to face. That's for sure. If we're not, I've got a meeting this afternoon to talk about how we're doing these labs remotely, because I don't think anybody's really happy with doing Zoom labs. They kind of end up being lectures, which is not what lab is supposed to be. So things are probably going to be changing one way or another, but they're definitely up in the air right now. Okay. Yeah, I think a lot of students are ready to go back in the classroom.
Question, Professor.
Yeah.
So with these labs being through Zoom, is there anything like that we are going to need to get on our own in order to complete the labs, or to like really kind of do it? I guess is the question or concern for me. Just because if it was in person, if there's things that we don't have at home.
Right. That is something that this afternoon's meeting is going to address.
Okay.
It's some labs are easier to do at home. For like the biology 1 classes, they have kits and they go to campus and pick up a kit of materials and then do the labs at home. But for physiology, a lot of the stuff that we do requires specialized equipment. And that's obviously not something we can have you guys come and check out.
Yeah.
We're talking about several different things. Ideas that have come up is having, well, this is a small lab. I don't have any problem with having all of us in there, but maybe staggering students coming in or something along those lines. But, you know, getting the administration on board plus all of the faculty on board is a challenge. But I will keep you guys in the loop. This decision came down, or I heard about it this morning. So we're still not really sure exactly what's going to happen.
Okay.
I'll post announcements on Canvas if I learn anything new.
Okay, thank you.
You're welcome. Could we do them outside? Probably not because of the equipment needs, but the nice thing about the labs on our campus is the rooms are large. They've got high ceilings and they have multiple doors. So we can open the doors and get good ventilation. And the ventilation system was just completely redone last summer. So it's brand new and it's much better than what you have in a lot of places. Personally, I feel like for the number of students we have in this lab, we could do the labs in person and be safe. That's my opinion. But yeah, we're still kind of trying to figure all of this stuff out. All right. So there is some reading on Canvas for you guys to do as your lab assignment for the week. Read that assignment and there's a quiz on that material. And just take the quiz on Canvas before next Tuesday, January 25th, before midnight. And it's just a short quiz. Okay, so this lab is about cells. If we were in the lab, we'd all have microscopes and we'd be looking at slides. And you would have had reading to do beforehand, but we're going to have to kind of go over this as a class. So when we're talking about cells, all cells have to have critical functions that are listed here. Regulating material that comes in and goes out of the cell. Converting food into cellular energy You probably learned about this already. This is primarily done in the mitochondria. Plants, of course, do photosynthesis. That's what we're talking about with converting food into cellular energy here. Cells also have to manufacture things like proteins and carbohydrates and lipids. So you guys are probably used to seeing little cartoon drawings of a cell that has a nucleus and maybe a couple of mitochondria, and it's all really simple. This is a false color electron micrograph of a real eukaryotic cell. It's only part of the cell. So this is the cell membrane, and you can see proteins here that are embedded in the membrane, a whole bunch of extracellular materials. These little squigglies are carbohydrates that are attached to the outside of the cell. And you can see all of these colored things in here inside. That's largely proteins. We've got a vesicle over here. And oops, we've got parts of the cytoskeleton coming in from various angles here. So I want to show you this because it's very easy to think about cells as those little drawings and not realize just how much is going on and how much stuff there is. So when we talk about glucose being converted to pyruvate in the cytoplasm, it's not one glucose molecule. It's tens, hundreds of thousands, probably millions of glucose molecules, all happening at once. We're talking about the cells because cell function is critical for human health. When the cells don't function, the body doesn't function correctly. We can talk about this in regards to diabetes. Diabetes is a problem with cells not functioning correctly. We can think about this in terms of cancer. Cancer is uncontrolled cell division. Cells are reproducing out of control. Now, you probably know this. There are two major types of cells, prokaryotic and eukaryotic cells. Prokaryotic, pro means before, and karyon refers to the nucleus. So these are cells that evolved before the development of the nucleus. They do not have a nucleus. And that includes bacteria and archaea. These are going to be primarily single-celled organisms. They can form colonies, they can clump together for various reasons, but they're not complex multicellular life like our bodies are. The eukaryotic cells, that EU means true. So these cells have a true nucleus. They also have membrane-bound organelles, and eukaryotic cells can either be single-celled, but we're really interested in them because they form complex multicellular life, like us. Also plants and fungi, but we're focusing on the human body. So we're really going to think about the human cell. So these are probably the images that you're more familiar with when we talk about cells. Nice, simplified, not to scale, definitely incomplete as far as how much stuff is in there. The Canvas reading that you're going to be doing talks about the different organelles and a little bit about their functions and construction. So we're not going to talk about that part today. What we're going to talk about, oh, I did want to show you this first. We're going to talk about primarily about cell division. Human cells come in a lot of different shapes and sizes. So they're not all nice, neat little circles. Muscle cells tend to be elongated and stretched out. Red blood cells are these unique little disc shapes. Nerve cells have all these projections sticking off of them. We estimate that there are between 30 and 40 trillion cells in the adult human body. You don't have to know this number, but I'm trying to blow your mind here because this is a huge number. We kind of have an idea what a million is. A million seconds is 12 days worth of seconds. Okay, that's pretty big. A billion seconds is 32 freaking years. A trillion seconds would be over 31,000 years. So when we say we have 30 to 40 trillion cells in an adult body, that's really an incomprehensibly large number. Okay. Cell, let me find my little annotation button. Cell cycle, cell division. These are both terms for one cell dividing and becoming two different cells. It is a highly, highly regulated process. It's one of the most important things to remember about all of this is that it is highly regulated and that regulation is very important. If something goes wrong with cell cycle, it can turn those cells into cancer. Different cells in the body divide at different rates. Neurons seldom divide after infancy, but skin cells divide very, very frequently. You replace your skin constantly. The digestive tract, you replace those cells constantly. Why do you think we have to replace cells in the digestive tract frequently? Quiet. Okay.
We're like using them so often.
That's part of it. Using them a lot, that's part of it, but it's also that the cells in our digestive tract are exposed to foreign material.
So they're kind of getting broken down in a sense. So they need to be replaced.
Yeah, they're getting damaged. So you take in food, that's foreign material. And yes, we break the food down and that's all good. But sometimes you bring in bacteria or you bring in compounds that are damaging to cells. And so those cells tend to get worn out faster. Neurons, they're nice and safe in here. They don't get exposed to very much. So when we talk about cell division, of course, we need to think about mitosis, right? I'm sure you've heard of this before. Now, one of the most common misunderstandings is that mitosis is the same thing as cell division. It's not. It is part of cell division. Specifically, the division of the nucleus. When we started looking at cell division, we looked at cells under the microscope. We divided this process up into stages. So these are all plant cells. Does anybody know how I can tell they're plant cells?
Cell wall?
The cell wall, yeah, exactly. They've got this rigid, almost square or rectangular looking cell wall. So when you look at these images, this looks like something very different from this one, which looks different from this one, which looks different from this one. You can see that under the microscope pretty easily. So that's how we divided up mitosis into those stages. And as we talk about the stages of mitosis, you'll see that they're not, they kind of blend together. Trying to figure out where one ends and the next begins can be a real challenge. This is true in a lot of biology. Trying to figure out if you see a spectrum of color, where does the color stop being green and start being blue? There's not a distinct line there. And amazingly, we can now see videos of this. Why is it not going to the link? Hang on, I've got to go to. All right. Yeah, go away. So this is going to show us a movie of actual cell division going on. So you can see things are moving around. Oh, look, all the chromosomes are lined up and they're pulled apart. Now this is a speeded up process. For human cells, it generally takes more like several hours for this process to happen. But what I hope you can see from this is how smoothly the transitions are between the different stages. So it's easy to see the stages, but it's not always easy to see where they begin and end. Okay. So this is the process of cell division, or the cell cycle. Generally speaking, if your cell is not actively dividing, it is hanging out over here in what we call G0. There's nothing going on except normal functions. Your islets of Langerhans cells are producing insulin. Your neurons are communicating with each other. Your liver is producing alcohol dehydrogenase to break down alcohol in your bloodstream. All of that stuff happens in G0. It is part of interphase. How familiar are you guys with this?
With the processes?
Yeah, with all of cell division and mitosis.
Somewhat. I mean, I just came out of anatomy, so.
Somewhat. I don't want to bore you by repeating stuff if you really understand it already. But since nobody's chiming in.
I think you're all good to go over it again. I think we could all use a bit of a refresher.
Okay, good. Then I will do that. Okay. Interphase is the part of the cell cycle that is basically not mitosis. And there are several subphases. So the G1 phase is post cell division growth. And if the cell doesn't need to divide again right away, it'll go into G0, normal cell function. And cells like your neurons, they spend most of your life in G0. They're just sitting there. And you see this little stop sign? That is, let me go write this over here. G1 checkpoint. So a checkpoint, and there are a lot more of them than are shown here. A checkpoint is the cell checking if it should go on and divide. So if your cell's sitting here in G0, it's just going to stay in G0 unless it gets a signal to divide. So it needs a signal to exit G0. And if it does that, if it does get that signal, it will go on to S, which is growth and DNA replication. Copying that DNA, making a second copy of all of the chromosomes. Followed by a few more final preparations in G2. More growth, storing nutrients and energy that it's going to need for cell division. And then we have the G2 checkpoint. This is a checkpoint where a protein called P53 is important. Has anybody heard of P53? No.
I have.
You have?
Yes.
Okay,
We very, very lightly learned about it in my very first biology class.
okay,
So I don't know a whole lot about it, but I have heard of it, yes.
okay. You've heard of it. That's good. That's better than probably anybody else in the class. P53 is a protein, and its job is to check the DNA for physical damage, and if possible, to repair it. So P53 works at this checkpoint to make sure the DNA, not to see if there are mutations, but to see if there's physical damage to the chromosomes. So a double-stranded DNA break, for example.
I have a question. Is it possible? You said if possible to repair it. So can it go to that checkpoint and not be repaired, like the DNA?
Yeah, so if it cannot repair the DNA, you're going right on to the next point. DNA cannot be repaired. P53 triggers a process called apoptosis.
That's the like programmed cell death, right?
That's programmed cell death. Yes.
So there's no way like the DNA would make it out of the G2 checkpoint without either being repaired or dying.
In an ideal world, yes.
It's one of those things where it can happen.
Yes. If you've got a genetic mutation in the gene for P53, it's not going to do its job, and the cell might go past this checkpoint and divide. That's cancer. So if your P53 is not working correctly,
Okay.
that's one of the most important genes in cancer.
Is the P53?
Is the P53 gene. Yeah. It's mutated in incredibly high percentages of, well, a lot of different cancers. I think lung cancer is one that it's most commonly damaged in. Most lung cancers have damage there. So there are other checkpoints. I'm mentioning just a couple of them, and I'm mentioning P53 because of its role in cancer. I know a lot of you guys are going into health fields, so these are the kinds of things that you'll probably learn more about. Okay, but if everything's good and P53 says we're good, the DNA is working, it's all looking great, then the cell goes into mitosis. All right. And here's our little apoptosis image. All kinds of weird things happen in apoptosis, but we're not going to go into the details of that one. You guys all did get that definition. I know somebody else said it for me. So let me make sure it's clear. Apoptosis is programmed cell death. It's designed to destroy the cell without damaging the organism. So a lot of things that are involved in this are things like making sure the enzymes in the lysosome don't escape and start digesting the neighboring cells. All right. You guys seen one of these before? Anybody know what it is?
Not like
These
that.
are chromosomes.
They are. They're human chromosomes, human male. And you can see, and I'm sure you guys remember all of this, but I'm going to go over the highlights. Most of the time, our cells have 2 copies of each chromosome. So we've got 2 copies of chromosome 1, 2 copies of chromosome 2, blah, 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 etc. This is called diploid or 2n, where n is the number of chromosomes. It's not a real number. It stands for the number of chromosomes. If we're talking about horses, I think they have 48 pairs of chromosomes. We have 23. So different animals have different numbers of different chromosomes. The entire collection of our genes is the genome. For reproduction, we need 1 copy of each chromosome per gamete. Gamete just means sperm or egg. And that's haploid. So when we look at diploid and haploid, or N, DI means 2. Ploid refers to number of chromosomes. When we name chromosomes, the largest chromosome is always named number 1. The second largest chromosome is number 2. The third largest chromosome is number 3. And we use the word homologous pair for the 2 copies of one type of chromosome. So chromosome. So like we've got the 2 copies of chromosome 4 blocked out. That's a homologous pair. Anytime you see homo in science, it means the same. Now, when we talk about the 2 different copies of the same chromosome, they're going to have the same genes in the same locations. So in this fictional little chromosome here, there's a gene way up at this end for fur color, a gene for eye color, and a gene for hemoglobin A. Hemoglobin is a protein that is involved in carrying oxygen in our red blood cells. But there can be different alleles. You guys heard this word before? Yeah. Does anybody
Yes.
want to give me a definition?
I think alleles are kind of like not the code for things, but like the label for them.
That's not how I would put it. This is one of those things that I have found students struggle with this word. Obviously, you guys have all heard it. Only Brenna was brave enough to venture forth a definition. Alleles are different versions of a gene. You can also think of them as different, oh God, I've got to spell recipes. Recipes for a protein. So up here, you've got a gene that is going to code for fur color. And if you think about dogs and cats and horses or people, our hair is basically fur. It can come in a bunch of different colors. And so this gene makes a protein that contains a color, the color fur. And in my example, it can be blue or it can be red. These are the same gene, but they are different alleles of the same gene.
So the alleles, like the variations.
It's the variation. Yep. It's easy to think about this with color because it's something that we can visualize, but lots of different genes have it. In us, most humans have one allele, the same allele for hemoglobin. But there is another allele that's not very common, and that's the sickle cell allele. If you've ever done some basic genetics and you've done Punnett squares, they're all about the different alleles. So if you've got an allele for sickle cell, you're either a carrier for sickle cell anemia or you have sickle cell anemia, depending on whether you've got one or two of these alleles. The other allele is normal hemoglobin. Does this help you guys understand it?
So is sickle cell, is that a dominant trait then? Or is it recessive since you have to have both?
Recessive until you take a genetics course. And then you'll understand, then you'll learn that dominant and recessive, it might apply to pea plants, but when it comes to people, it's much more complicated than that. But yeah, you're on the right track there as far as the alleles definition goes. Okay. All right. I'm sure you guys know an individual strand of DNA forms a double helix. You probably vaguely remember that they're connected by complementary nucleic acids or bases. Adenine binds to thymine, cytosine to guanine, ringing some bells in the back of the mind. Awesome. Now, when we talk about chromosomes, though, over half of the chromosome is actually made of proteins. These proteins, one of the most common one are these histone proteins. And they're involved in making DNA into the stick-like chromosomes that we think of. So when our cells are doing their normal thing, DNA has kind of a loose structure to it. But when it's time for cells to divide, we get those long stick-like looking structures, what we can see under the microscope. And the histones are really important. I mean, they're important for normal maintenance of the DNA, but when it gets into this chromosome shape, the histones are really important for coiling up the DNA really, really tightly.
And the histones are a type of protein?
Yes, the histones are a protein. And this stick-like form of DNA is called condensed. And the looser one, yeah, well, that's just relaxed. That's an easy one to remember. And the relaxed form is when RNA is actively being made. So normal cell function, if you're making messenger RNA, it has to be in the relaxed form. You can't, oh God, here we go again. You can't make RNA when it's in the condensed form. And when you take genetics and you find out that I was wrong, there are exceptions to every rule. I hate to say never. This is one of those cases. Most genes cannot be converted to messenger RNA in the condensed form. All right. Did I hear somebody unmuting?
It was me, but I remembered what you wrote, that's all.
Okay. So there's a little bit more I want to go over, and then we're going to do some activities, finally. Okay. Most of the process that we're going to be talking about is mitosis. There's some prep work, like we said, and then after mitosis is the process of cytokinesis. Cyto, anytime you see cyto, it refers to cell. So cytokinesis is the actual division of the cell. So the first thing that happens once the cell gets a signal to start dividing, it's going to duplicate the chromosomes. I'm going to duplicates the chromosomes. I'm marking out a whole bunch of stuff because this is supposed to be lab, not lecture. This is an actual electron micrograph of the 2 copies of the same chromosome that are still connected to each other. And the different stages, we duplicate our chromosomes, the 2 identical copies stick together, and then they line up in the middle of the cell, and they get pulled apart to the opposite poles, and then the nuclei gets reformed. So let's go through that a little slower, looking at actual microscope images. This is prophase. You can tell it's prophase because the chromosomes are condensed. They're dark, you know, that almost stick-like looking thing. You don't see a clear nucleus. And this is a great time for being able to count the chromosomes. It's a really hard thing to do until you get a lot of practice at it. Then the chromosomes line up in the center of the cell. So you can kind of see that they're lining up here, right in the middle. That's metaphase. And this is called the metaphase plate. It's just a fancy word for where all the chromosomes are during metaphase. And anaphase, the chromosomes are being pulled to opposite ends of the cell. So they look kind of like, okay, I'm a biologist. They look kind of like, you know, spiders being pulled or octopi being pulled to opposite ends of the cell. They're being pulled kind of by the center of the chromosome, and the ends of the chromosome just kind of get dragged behind. And then you get to telophase. So this is telophase in an animal cell. And what you can kind of see going on here, you've still got the chromosomes a little bit condensed, but they're starting to relax and lose that stick-like appearance. And we're starting to see the sides of the cell indent in preparation for cytokinesis. So this is telophase. Telophase does not last very long and is often really hard to find examples of if you're looking at the cells under a microscope. This is cytokinesis, where the cell membrane, it shrinks down in the center until it pinches off, and it will eventually pinch off and produce 2 separate cells. Plants and fungi do this very, very differently. We're not going to worry about them. What I do want to mention is that sometimes mitosis occurs, but cell division doesn't, the cytokinesis doesn't. Mitosis without cytokinesis, you end up with a cell with multiple nuclei. Now, not surprisingly, this is common in cancer, but it's common in some other cells in the body. Anybody want to come up with a wild guess why healthy cells in the body might need more than one nucleus?
Because they're working harder than other cells or
Oh.
doing more.
Fantastic. Yes. Yes, Brenna, you're exactly right. They have to make a lot of protein. So they need multiple copies of the genes so they can make all that protein. Specifically, where you tend to see this most is in muscle cells. So if you're looking at a cell under the microscope and it's a muscle and it has multiple nuclei, that is normal. It's also normal in the placenta during pregnancy. Normal muscles, placenta, abnormal cancer, and certain viral infections. A prime example of which is HIV. HIV can cause this to happen. And fun fact, the genes that allow the placenta to have multiple nuclei, we think we probably got those from way back before we had placentas from a virus. So we think we got the gene from a viral infection, and it allowed us to be able to give live birth. How's that for funky? All right, do I have the other one up? Okay. Do, 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 do. Okay. Oops. When we look at this image, can you guys tell which phase that the arrow is pointing at? Which phase of mitosis?
Anaphase.
Anaphase, exactly. How about, oops, I didn't mean to make a new arrow. How about that one?
Telophase?
Telophase, yeah, perfect. How about that one? That one's kind of tricky, isn't it? Huh?
Prophase?
Prophase, exactly, yeah. Good job. All right. Let's vote on this one. What phase do you guys think this one is in? Okay, we've only got half of y'all answering. Okay. All right. This is exactly what I was kind of hoping for, because it is kind of difficult to tell. So the chromosomes are kind of lined up here in the center, but it's not as obvious as the images I showed before. So this is either kind of at the very beginning of metaphase, kind of the end of prophase, or it's the end of metaphase transitioning into anaphase. You can't always tell, you can't always define this as a specific category. Okay. What about this one? Okay. Again, we had some disagreement here. This one is prophase. You can tell because you can see the chromosomes, and they're kind of distributed widely throughout the cell. They're not condensed in any particular part of the cell, and they're not like you can see in the next next cell here how they're kind of that stick-like shape. They haven't formed that one yet. So this is prophase. This is a plant cell again. Okay, we do still have a little bit of confusion, but yeah, this is really cytokinesis. You can see the new, this is actually a cell wall forming, but it's also in animal cells, this would be the plasma membrane forming between what are going to be the two new daughter cells. Oh, I didn't share the results there. How about this one? Okay, again, we're having a little trouble with this one, but it is anaphase. And I know some of you are probably cringing at this, but anaphase is the spiders or octopi, if you prefer, octopus. So the outer side of the cell, I'm going to draw over it for you. So here, that is one cell. And so you can see if it was metaphase, you would see all of this in the center, and there would just be one of these clusters that looks like this. Because you've got two, that means the chromosomes are pulling apart to opposite ends of the cell. This one's a little bit tricky. So I'm going to draw the outline of the cell so you guys are clear exactly what we're looking at here. That's one cell. And it didn't relaunch the poll, did it? There we go. Yes, this is telophase. The chromosomes have started to relax and they aren't as obvious in these ends. Okay. So where am I? It's a good question. Where am I? How are we doing with that? Starting to get the hang of it? It's quite complicated learning to look at things under the microscope. I really wish we could do this in lab because learning how to look through a microscope is a very valuable skill to have. All right. One more thing I want to do before we wrap up for today. And what I want to do is I want you guys to send you into breakout groups. And I want you to talk about stem cells. Have you heard of stem cells? Do you have an idea what they are? Where do we get them? It's a controversial topic in some circles. So, oh, I hate that. I'd like for you guys to chat for a few minutes. I'm just going to put you in 2 groups because we've only got 8 people here today. And you can talk about it, and in about 5 to 10 minutes, we'll come back and talk about it as a class. Okay, so one of the groups was being very quiet and the other one was talking about chemistry. So I guess you guys didn't have a lot to talk about when it comes to stem cells, huh?
Our group really didn't know a whole lot about them other than that they're kind of like a base cell, kind of. And they do a lot of harvesting from the spinal cord and bones, like bone marrow, sorry.
Yeah, bone marrow.
And I think placenta as well, but I'm not 100% sure on that one.
Okay.
I looked it up on Google and they were saying they get it from the umbilical cord, the umbilical cord and the blood, which I didn't know.
Mhm. Okay. Well, let's talk about stem cells. Okay, so we all know we start when a sperm and an egg combine and you have a fertilized egg. And that fertilized egg is capable of becoming all of the cells in the body. We call this, there are various terms, pluripotent or totipotent. Those, you don't have to write those terms down. But as that fertilized egg divides and becomes more complicated, the cells start to become at least partially specialized. And the first step is specializing into the major cell types: muscle cell, nerve cell, epithelial cell, and that sort of categories. And as you develop, your cells become more and more and more specialized. So a stem cell is any one of these cells up here that has flexibility in what it becomes. These cells, when we're adults, we have stem cells. We have them, for example, in our bone marrow. Those can produce all kinds of blood cells. They're not going to produce new neurons, so they're a little bit limited. But there's a lot of interest in using stem cells, especially from, for example, the umbilical cord, where there's a lot more flexibility in those cells because they haven't become specialized yet. Using these in medicine to repair damaged organs, neurological conditions, heart disease, diabetes. There's a lot of research that's going into this at this point. There are some treatments that use stem cells. Bone marrow transplants are an example of stem cells in medicine. And they can help repair all kinds of different blood diseases, for example. It's much more complex than I'm making it sound here. And the average person really doesn't understand it. There's a lot of criticism from people who don't understand what's going on or where these come from. The anti-abortion people will tell you that stem cells have to come from aborted fetuses. That's just not true. That's not where we get stem cells today. We can even, to some degree, induce cells from an adult body to become stem cells for research at this point, at least. And when we talk about stem cells, oh, nope, this is changing the topic, and I don't want to go on to the next topic. I think this is a good place to call it quits for the day, looking at puppies. All right. I have a meeting scheduled for this evening. I'm hoping to find out more about what we're going to do with this lab. I'm really hoping there are some better ideas than, kind of having you guys have to listen to me talk for an hour and a half. You didn't sign up for a lecture here. This is supposed to be hands-on stuff, but I'll have to let you know how that goes. Does anybody have any questions before we wrap it up today?
I don't know if you've repeated yourself with this question. If you did, I'm sorry. As far as the recorded, whatever these are, the lab lectures go, are they saved somewhere on Canvas? I haven't been able to find it.
I will double check and make sure I put a link in. They're going to be on YouTube.
Oh, okay. Okay.
And I'll double check and make sure the YouTube link is on Canvas and functional.
I did see the YouTube section on there. I just didn't think that that's where the recorded lectures were. Sorry.
Yeah, it's easier to put them on YouTube than to put them on Canvas. And YouTube, you can set it up for closed captioning, which you can't on Canvas.
Gotcha. Thank you.
You're welcome. All right, if there's nothing else, we'll call it a day.
Okay, have a good day.
Bye.
I did have a question. It was just more kind of specific to something that happened to me on Canvas with like a technical issue.
Okay.
I was kind of just going through this week's module and like looking at the notes for